Click the bell icon and subscribe to our channel so that you will not miss any classes. In this class, let us study the simple. In this class, let us study the microbiomolecules which are present in the acid soluble fraction, that is in filtrate. First, let us study about the simple sugars. In NCERT text, they have not given about simple sugars, but they have mentioned in the structure about glucose and ribose sugar. Now, the simple sugars in living systems are classified based on the number of carbons they have as triose sugar, tetrose sugar, pentose sugar and hexose sugar. And here this classification is mainly based on the number of carbons they have. Example, triose have 3 carbon, tetrose have 4 carbon, pentose have 5 carbon and hexose have 6 carbon. Let's try to write the examples for each. Example for triose sugar, the intermediate compounds formed in glycolysis, example phosphoglyceraldehyde and dihydroxyacetone phosphate are the examples. Example for pentose sugar are ribose sugar and deoxyribose sugar, which are present in DNA and RNA. Hexose sugar example is glucose. In your NCERT textbook, they have given the structure of ribose sugar and the glucose. Respectively, they are pentose sugar and hexose sugar. The structure of ribose sugar and glucose are given in page number 107. You can refer in NCERT textbook and it goes like this. The glucose structure is as it is a 6 carbon compound. The structure goes like this: OH, CH2, OH, here OH, here OH and here OH, and remaining all are H, that is understood. And the structure of ribose sugar is here. In studying about the DNA and RNA, we should come to know that there are 5 carbons starting from in the position 1, 2, 3, 4 and 5th carbon is of CH2OH, and here in both second and third carbon there is presence of OH group, and this is the structure of ribose sugar and the glucose. And that's all given in NCERT about simple sugars, so that you need to remember. And another type of microbiomolecules found in the acid soluble pool are amino acids. Amino acids are simply referred to as the compounds which are having amino group and the hydroxyl group which are attached to the alpha carbon. And here alpha carbon is having 4 groups attached to it, include carboxyl group, amino group and the R group, and of course OH group. So these are the 4 different groups which are attached to the alpha carbon. And here, since it resembles the structure of a methane, which is CH4 like this, and here 4 carbon hydrogens are replaced by 4 different groups, hence the amino acids are also referred to as substituted methanes. So this is given in NCERT, so you need to remember substituted methane, that is carbon which with the 4 hydrogen atoms. Here instead of 4 hydrogen atom, it has 4 different groups attached to them. They include carboxyl group, amino group, hydrogen and the replaceable R group. Now the R group is variable in different types of amino acids. Depending upon the type of R group, there are 20 different types of amino acids which are found in proteins. So here this point is very important. To form proteins, 20 different types of amino acids are responsible. Now these 20 types of amino acids are formed by the different R groups which are attached to the alpha carbon. If alpha carbon has the R group as hydrogen, then the amino acid is called glycine. Same, if the alpha carbon has the R group as methyl, that is CH3, then the amino acid is alanine. If the alpha carbon has R group as hydroxymethyl CH2OH, then the amino acid is serine. So these 3 structures are also given in NCERT, so you need to remember the structures of these also. Now, depending upon the carboxyl and amino group present in a amino acid. Amino acids are classified into three types. They are acidic amino acid, basic amino acids, and neutral amino acids. So basis, you have to remember, it is based on the amino group and the carboxyl group. Now example for acidic amino acid is glutamic acid. Basic amino acid is lysine. Neutral amino acid is valine. Same way, there are aromatic amino acids are also present which have a aromatic R group. Example, tyrosine, phenylalanine, and tryptophan. That is about the classification. And here the chemical and physical properties of amino acids, so how they behave chemically and physical nature, is purely based on the carboxyl group and amino group. So this they may ask, physical and chemical properties are not self, they are dependent upon the carboxyl group and the amino group which are present in them. And a particular chemical property of an amino acid is ionization. That is, amino acids are ionizable. This ionizable property is with respect to, again, carboxyl and amino group. And this ionization of carboxyl group and amino group is based on the pH of the surrounding in which the amino acid is present. For example, if an amino acid is placed, so I'll write the basic structure of amino acid first. Here NH2, C, COOH, H and R. If this is placed in a aqueous solution, then these two will undergo, that is carboxyl group and amino groups will undergo ionization to form COO minus C, NH2, and here R. Sorry, here it is NH3. And this has got both positive charge and negative charge. So this is the ionic form, and this ion is called bipolar or Zwitter ionic form. And when this Zwitter ion is placed in a acidic solution, then in acidic solution the concentration of H plus ions are more, hence this H plus ion will bind with a carboxyl ion to form COOH, C, H and NH3 plus, and here R, and the amino acid has got a positive charge. The same if it is placed in basic solution where OH minus ions are more, then this is basic solution, then the amino acid will got COO minus C, H, R, and here NH2 again with the release of water molecule, and now this has got a net negative charge. So which means the ionization property is purely based on the carboxyl group and amino group, and mainly with the pH of the surrounding environment where they are present. And the Zwitter ionic form is given in NCERT textbook, so you have to refer that and you have to remember. There they have mentioned B is the Zwitter ionic form, so you have to remember how this Zwitter ionic form is formed. It is formed by the donation of hydrogen from carboxyl group to the amino group, so that the amino acid will got both positive charge and as well negative charge, and that is called Zwitter ionic form.